aerobic metabolism. Those three are going to be on the quiz that opens today at 6 p.m. You have until the 26th to finish the quiz, right? So you have like 4 days to take the quiz. It's like 5 questions. There's like 10 or 15 minutes to answer it. So you have plenty of time to prepare for that if you need to. Yes.
Do you know yet if our first exam will be all essay?
I'm not 100% decided yet. Yeah, I'm leaning towards maybe a mix of multiple choice and short answer. Kind of leaning toward that, but we'll decide. Yeah, I don't know if it'll be all 100% essay. Kind of thought maybe that would be. To be honest, the essay part of the test usually helps people. In general, like, I give you more credit if you word it correctly versus even if it's not like 100%, like you spelled this wrong and like, well, I know you're meaning, right? So I tend to like work in people's favor for that written part. But having an entire exam full written choice, written essays would be might be challenging for some people. We'll see. I'll probably be maybe 2 or 3 questions, but not like the whole thing. Probably not. I thought maybe that would be fun, but not fun. But I thought it would be like interesting, because like that is a good way to assess people and how well they know it. But sometimes like multiple choice can be a little more efficient grading wise. So that's a lot to read. So. Also, usually with the short answers, when I do it like that, it'll be like a page, but it'll be like on a box or like half of the page, and you have to write what you're gonna write and fit it in that box. But sometimes when I did the essays the first time, people would like write as much as they possibly could, and it's like, well, now you're like taking away time from the actual multiple choice part of the exam where you didn't need to write all that to get the answer correct. So. There would definitely be like a dedicated area you can write in, so you'll have an idea about like how much is enough to be a complete answer in terms of like volume of text. Alrighty, so let us talk about the aerobic metabolism. So we'll finish this and then continue on. So if we just go back, we left off on slide 14, but we'll go back just 2. So if we think about the regulation of the Krebs cycle, there's a couple enzymes in the Krebs cycle that regulate how quickly the Krebs cycle is going through oxidation. Okay, so one key site of regulation would be citrate synthase. Okay, this enzyme synthesizes citrate. Okay, what is citrate? Citrate is oxaloacetate here, which is the end product of the Krebs cycle. It joins with acetyl-CoA, and with the enzyme citrate synthase, you form citrate, right? So citrate is the first product of the first molecule that's produced in the Krebs cycle, the start of the Krebs cycle. So citrate synthase is regulated. Okay, we also see that isocitrate dehydrogenase is regulated, and then so is alpha ketoglutarate dehydrogenase. So those 3 sites are regulated. Now, if we see what are they regulated by, well, ATP and NADH and succinyl-CoA inhibit citrate synthase. So if we go back here, if we're saying a high concentration of ATP inhibits citrate synthase, why do you think that would inhibit citrate synthase?
Because you don't need to make ATP.
Exactly. The point of the Krebs cycle is to produce hydrogen ions through the oxidation of these substrates. Okay, those hydrogen ions should go to the electron transport chain to produce ATP. So if you already have a lot of ATP, there's really no need to kickstart the Krebs cycle to make more citrate and have that cycle speed up, because there's not a need for that cycle to even be active in the first place if there's already a lot of ATP. The other thing that inhibits this pathway inhibits that enzyme is NADH. Okay. Remember that in the Krebs cycle, if we're also including the intermediate step, notice you need NAD plus here, you need NAD plus here, here, and here. So you either have NAD plus or you have NADH, right? So it's either oxidized or reduced in that form. So if you need the NAD plus and all you have is NADH, look at all these steps in the Krebs cycle that you can't have those steps occur, right? So it's like, okay, maybe we produce citrate, but then when we get to the isocitrate dehydrogenase step, we don't have enough NAD plus, so now we've made citrate for no reason, right? We can't do anything with it, okay? Or maybe we make alpha ketoglutarate and there's even less NAD plus, so now we can't make succinyl-CoA, right? Or we make succinyl-CoA. If we make the succinyl-CoA, but then we don't later on in the Krebs cycle don't have enough NAD plus, now we have a succinyl-CoA that's kind of built up and it's not being processed into oxaloacetate. Okay, so these are all just examples to show that it's these sites of regulation can feed back to the start of the Krebs cycle to slow it down, because whatever is happening within that Krebs cycle is not sufficient to maintain the speed of that cycle. Okay. Isocitrate dehydrogenase is the rate limiting enzyme of this system. Okay, so ATP inhibits this enzyme. ADP and NAD plus activate it. Okay, so notice how in this step you need an NAD plus for this step to happen. Notice how, like, if NAD plus is available, that actually turns on the activity of isocitrate dehydrogenase. Okay. If you don't have NAD plus available, now here's this signal that is supposed to stimulate this enzyme is not there anymore, right? So if it's not being stimulated, it's not as active, right? So how can we increase the rate of this pathway? We don't have it being stimulated correctly. It's not, right? Now what's interesting actually, if you think about this, also notice that ATP inhibits it and ADP activates it. So during exercise, we are using ATP, correct? Right, so the inhibition right here, ATP, that would actually go away during exercise, right? So if the inhibition goes away, does that mean isocitrate dehydrogenase can be more active? Yes. Also, when you break down ATP, you're producing ADP and inorganic phosphate. Notice ADP stimulates that enzyme to be active. So just the process of you breaking down ATP removes the inhibition and stimulates that enzyme to be more active. So notice how it's causing that shift. But unlike glycolysis, where you break down ATP and then you break down phosphocreatine, right? And then you can then that activates PFK and so on and so forth. Notice how that NAD plus is required to activate it, right? And also that step, this step here, requires NAD plus. So even if you have a large demand for ATP because you've broken down ATP and there's a lot of ADP, the fact that you don't have NAD plus, that's something that's like, oh, it's stimulated because of ADP, but it's not maximally stimulated because you don't have enough NAD plus. Does that make sense? So in a perfect world, this enzyme would be highly active if you've broken down enough ATP, built up enough ADP, and still had enough NAD plus, right? Then that would all indicate very positive environmental stimuli to make that enzyme be really highly active, right? You could have a lot of ADP, but if there's no NAD plus, we're not maximally stimulating that enzyme, right? So the enzyme activity goes down, and also whatever that enzyme would normally be doing, its activity is a little lower. There's not even that substrate to make that reaction occur, right? Like this step here requires this molecule. So by not having this molecule and only having NADH, that actually reduces the activity of this. This activity is now lower, and also you can't even convert that into this because there's no NAD plus, right? And also key thing here, if we don't have NAD plus, low activity, that means we only have, or we have a lot of NADH, but what does this do? This feeds back to citrate synthase to slow down that activity, right? Because doesn't NADH inhibit citrate synthase activity? It does, right? So this is now they get to the step we've made citrate. Okay, we just maybe need some more ATP, right? ATP is really high. Oh, there's not enough NAD plus. Can't have this step occur. Activity goes down, feeds back to citrate synthase. Let's stop making citrate because even if we make it, we can't do anything with it, right? Notice the point of the Krebs cycle is to produce these hydrogen ions and then they go to the electron transport chain. If we've made a citrate, but then we get to this step and we can't make a hydrogen and use anything with it, essentially we've made a substrate we can't make any energy from. There's no hydrogens produced in these steps here, right? So there's nothing going on. Also remember, the intermediate step requires NAD+. So we don't have NAD+ here. Certainly we would need to start slowing down this step too, because the whole point of this intermediate step is to make acetyl-CoA, to go into the Krebs cycle to make the here. And remember, what regulates the conversion of pyruvate to acetyl-CoA? PDH. What affects PDH activity?
How much pyruvate we have?
PDH phosphatase and PDH kinase. What regulates their activity? Exactly, right? The ratio of how many ATP to ADP do you have, and then how much available NAD plus to NADH available. Remember, having a low NAD to NADH ratio means I have this many NAD plus and this many NADH. If that ratio is really low, that means that the PDH kinase is going to be really active. What would that do to the conversion of pyruvate to acetyl-CoA?
Inhibit.
Slow it down, right? Kinase activity goes up, conversion of pyruvate to acetyl-CoA goes down. Right? So now we can see, like in the Krebs cycle, just not having enough NAD plus slows down the rate limiting enzyme. We know what rate limiting enzymes do. Slows down the conversion of the activity of citrate synthase, which reduces the product of citrate. Okay, now we have the less starting product. And now it feeds back even into the intermediate step to slow down whatever's even going into the Krebs cycle, right? So this is all factors, these are all factors that says, hey, we're shutting down the aerobic system because we can make any kind of substrate and put it in there and we can't do anything with it. There's no way to make energy because there's not enough of these. So what can we do? We'll shift to glycolysis. And with the presence of which enzyme? What enzyme allows us to make ATP anaerobically?
PFK.
Well, PFK is a regulating enzyme of glycolysis, right? But say we get to the end of glycolysis, we produce some ATP, we have a pyruvate. There's a key step in glycolysis that requires NAD+, right? Remember, we invest ATP, and then after we split that molecule, there's this NAD+ dependent step, and then we make ATP after that, right? This NAD+ dependent step is required to have a net positive ATP production, right, in those following steps. If we don't have enough NAD+, that means the Krebs cycle shuts down, so we're not making ATP aerobically. But also, if there's no NAD+ here, then there must not be enough NAD+ in the cytoplasm as well, in glycolysis. So if we don't have any in glycolysis either, how can we make ATP anaerobically? Exactly. Remember, lactate dehydrogenase, normally you need this to make the ATP. If you only have these, luckily lactate dehydrogenase can take the hydrogen off of this molecule, put it onto pyruvate. Now we make lactate, and now we have an NAD+ to make ATP anaerobically, which is something we can't do in the Krebs cycle. Right, so does this help you understand why we can only use carbohydrates to produce ATP in glycolysis? Because even if we have that carb, we can use it anaerobically, but once we turn it into an acetyl-CoA, we can't do anything. There's no way to make energy from that, right? And fats get turned into acetyl-CoA, which get put to the Krebs cycle. So we can't use those aerobically either for energy because fats are acetyl-CoAs. They can't be turned into a form like carbohydrates, right? So this all relates back to why only carbohydrates can be used anaerobically in high intensity exercise. Cool? All right, so that concept's important because on your exam, you know, I know we talk about these concepts like this lecture and this lecture and this lecture, but on the exam, the questions are actually incorporating components of different lectures into the same question. So you might get a question like, oh, in which type of exercise intensity are you going to maximally use carbohydrates, and what's the enzyme that regulates that process, right? And I could give you PFK, I could give you hexokinase, I could give you glycogen phosphorylase, right? I wouldn't put PFK and phosphorylase in the same answer because they actually both would be the right answer, right? But if I gave you, you know, high intensity activity, maximal intensity activity, 100% VO2 max, what substrate are you using? Blood glucose or glycogen?
Can you say the question one more time?
So if you were exercising at 100% of your VO2 max, what substrate are we going to use? Blood glucose or glycogen?
Glycogen initially.
Glycogen, right? The muscle uses whatever substrate's located locally. For you to use blood glucose, you have to first make sure there's enough blood glucose in your system. Then it has to be transported into the muscle. And realistically, you have way more glucose stored as glycogen than you do the availability in your blood glucose stores, right? The liver maintains for all the tissues, but what we need for exercise is only really in the muscle. If you're exercising for an hour, 2 hours, 3 hours, then blood glucose becomes a good, what's maintaining the replacement from, you know, in those stores, right? But initially, max exercise, we're going to use primarily glycogen. So, which enzyme regulates that process? Hexokinase or glycogen phosphorylase? Glycogen phosphorylase, right? So we can see, like, okay, if I have fats, glycogen, okay, which substrate? You got to get that right. If I said blood glucose, glycogen, which enzyme, hexokinase or phosphorylase, right? It's phosphorylase. So notice how they're from different lectures, but they're related to the same concept. Okay, so try to see how we're trying these together. But this is a good example, right? Not enough NAD plus. Okay, we got to shift back to anaerobic metabolism, lactate production, glycolysis. Right, and then alpha ketoglutarate. Dehydrogenase, notice that NADH and succinyl-CoA also inhibit that activity, and AMP activates it. So just like these other enzymes, when you start to use energy, that stimulates its activity, hence AMP. There's not enough NAD plus available because you only have NADH, shuts it down. If you have a lot of succinyl-CoA, that means succinyl-CoA, remember, if you need that substrate, it should be processed into a product. If that product is not getting used, either the enzyme's inhibited or there's not a need for it, right? So now we need to shut down the production of that, and it slows down the Krebs cycle, okay? So, and then this kind of just reiterates that I circled that right there. All right, so we can use fats for energy, okay? Fats are stored as triglycerides. For us to use those fats for energy, we have to undergo lipolysis, and those triglycerides have to be converted into a glycerol and free fatty acids, right? Normally on a triglyceride, remember from lecture 1, we have a glycerol molecule and 3 fatty acids that are attached to that molecule. Lipolysis breaks those fatty acids off. Now they're free, they can go into circulation, they can go into the mitochondria, you can make energy from them, okay? Those free fatty acids are then converted into acetyl-CoA in a process called beta oxidation. Okay, so don't confuse the terms lipolysis and beta oxidation on the exam because I ask you about specific components of each. Okay, I've had students that they knew beta oxidation, they were just calling it lipolysis, right? So they got those questions wrong, but conceptually they knew that material. Okay, so make sure you understand that lipolysis is literally breaking down the fats and then liberating the free fatty acids. Once they're a free fatty acid, those fatty acids are then going to be converted into acetyl-CoA in a process called beta oxidation, right? And we don't primarily use glycerol for energy because it's just not a substrate we use. We're going to get the majority of it from the fats and the carbohydrates, the fatty acids and the carbohydrates. Protein. We do use protein for energy. It's got to be, we have to first break down the amino acids. We have to strip it from the nitrogen, right? The nitrogen comes off, then we kind of have this carbon backbone that can be used like glucose or pyruvate or converted to acetyl-CoA or put into a molecule very similar to a Krebs cycle intermediate. But that entire process requires oxygen. Okay, so even though we can get the same number of calories from protein for energy, right? Carbs, 4 calories. Proteins, 4 calories. Even though we get the same energy from it, it actually requires more oxygen to get there, right? So we can only use carbohydrates anaerobically. We cannot use proteins. And then back to, lecture 1, proteins we're probably going to use if you're on a diet, calorically restricted, starvation, really long duration exercise, like 3 hours or more. In those conditions, then you're going to be using a lot more protein. In a exercise intervention or intensity that's like 65% VO2 max for an hour, no. 10 sets of 10 reps of five sets leg press, no. Anaerobic glycolysis. You can be done with that in a half an hour. All right, so we're not going to use the protein for energy in a resistance exercise. And we're only going to use protein for energy with aerobic exercise if it's very, very long duration, like 3 hours or more. Okay, and even then it's no more than 10% of total expenditure. So, and I say that because on the exam, options, what are we going to use? Proteins or this or that, right? Proteins and amino acids will be an option on the exam. Don't choose that. Or if I do say under starvation or under caloric deficit or exercise more than 3 hours, then that would be the answer, right? But the protein itself wouldn't be like, oh, that's the one we're using, because we don't really use it. So when we talk about making energy, the whole hypothesis around how we produce ATP with these hydrogens in the electron transport chain are based around the chemiosmosis hypothesis. So essentially, these chaperone molecules like NADH and FADH, they go to the electron transport chain, they shuttle their hydrogens into the inner membrane space, right? There's a higher concentration of hydrogen ions up here, and they naturally want to fall through this enzyme called ATP synthase, okay? On this image here, ATP synthase is the enzyme that phosphorylates ADP into ATP, okay? So we know, a lot of physiology is based on compartments, right? Your cells are compartmentalized. When you have a compartment or a membrane, you can put a higher concentration of things on one side and a lower concentration on the other. And that difference in concentration is actually a way for cells to do work, right? Because they want to fall from high concentration to low concentration. It's doing the work for you, kind of like, a rotary and waterfall, that whole thing. So what happens is you have this high concentration of hydrogens in here and not out here, so they naturally want to come through. However, when there's oxygen inside this matrix, it is like a magnet. It really pulls those hydrogens through out of the inner membrane space and into the matrix to form water and ATP. Okay, so why does the electron transport chain, why do they call it oxidative phosphorylation? Because when there's oxygen here, the hydrogen are like magnetically, they're attracted to that oxygen, they are pulled through to form water. And when they're pulled through, think of this like a rotor. You go to the subway and you know how there's that thing that you have to step through, you swipe your card and that thing that rotates, you have to like really squeeze through it and it turns, and it's like a turnstile or something, I think they call it. So imagine like you're gonna go through this on subway, you splash your card and okay, I'm good to go, and you have to really squeeze through to get through, right? And that thing turns and it lets you pass through. You are the hydrogen, okay? So you are passing through that gateway that has this turnstile on it, right? Imagine that turnstile has an ADP and an inorganic phosphate. And as you go through that entrance, as you turn it, your presence of passing through it is what's physically turning that rotor, right? So as you turn that rotor, so as ATP synthase turns, it's literally putting that ADP and inorganic phosphate together. So you go, pass through, puts it together. Now we've made an ATP, right? So it physically phosphorylates that molecule, Because hydrogen has passed through ATP synthase. Now, why did hydrogen pass through ATP synthase?
High low concentration.
Because it's attracted to the oxygen, right? If there's no oxygen down here, like doesn't, you have two, positive, negative magnets. If they're far away from each other, do they move? No. If they're close enough, they get drawn to each other, right? So if there's oxygen here, the hydrogens want to come through ATP synthase. They want to pass through, they form water, and in that process, then they produce ATP. But if there's no oxygen here, they don't pass through. So if they don't pass through, are we making ATP? No, because what's turning this enzyme, this motor? Nothing, right? So that's why we cannot produce ATP without oxygen in the electron transport chain, because if oxygen is not present, that ATP synthase doesn't work. And so, like the enzyme says, it synthesizes ATP. If it's not working, it's not synthesizing, it's not making ATP. Now notice, you have NADH here. It's kind of like, I like to use that subway example because I think it's pretty apt. Imagine this is like a subway car, right? You're inside the subway car. It's hot, it's rush hour, it's filled with people, right? You can only fill that subway car with so many people, right? If it's full, you gotta wait for the next stop. You gotta wait for the next train to come by, right? You can't do anything, you're stuck at that stop, right? Imagine you're trying to get on that train, but it's already saturated with hydrogen, it's already saturated with people. You can't drop off more hydrogens unless this has actually made some space for it and kind of cleared, right? So if this is saturated, this cannot drop off, right? If we're stuck holding on to NADH, we can't get rid of it, right? So does this relate to the Krebs cycle? Yeah. If we can't get rid of this, then in the Krebs cycle, we don't have NAD+. In glycolysis, we don't have NAD+. Okay, and so because we're stuck with these hydrogens, because we're saturated up here, and we're saturated up here because there's no oxygen to clear that space, right? And you're not making, you don't have oxygen, you're also not making ATP. So this whole thing suggests, oh snap, I guess we gotta go back to glycolysis, where with glycolysis, we can use lactate dehydrogenase to take the hydrogen off and make lactate and then make the ATP there. It's not a lot of ATP, but we can make it very quickly.
Oh, so if NADH can't go through there, it's gonna go through lactate dehydrogenase?
Yeah, so it has the hydrogen, right? Once it's there, the only way to get rid of it is got to go to the ETC and drop off the electrons and pass them through the cytochromes, right? If it gets here and it's already saturated, the difference doesn't let it actually pass it through. So now it's stuck holding on to this hydrogen, okay? And it can't, there's not enough oxygen, this stays saturated, right? So you gotta wait, you gotta wait, you gotta wait. What happens when you go at rest, right? Let's say you're working out really hard, you're running, and then you slow down. What's happening? The work rate slows down, the muscles are not contracting as fast, you use less oxygen, you use less ATP. As you slow down, the demand for oxygen goes down, your ventilation's still up, you get more oxygen. Right? So just imagine you're working out really hard, then you literally consciously slow down your intensity, you lower the work intensity, you can supply more oxygen, right? When you supply more oxygen, that allows you to make more ATP to shuttle those hydrogens away, and now you can be in steady state, right? You slow down, you work out, you speed up again, harder exercise, not enough oxygen to supply that energy. If you're using more ATP, remember, every time you hydrolyze an ATP, you produce a hydrogen. So every time you're splitting molecules, you're producing hydrogens. And these NAD pluses go in and grab them because they increase acidity. And also, we can use them to make energy. You go through glycolysis, how many steps require produce an NAD plus? They do, there's a few of them, right? The intermediate step, right? All those steps produce these hydrogens, right? So these are going to be accumulating very quickly, okay? And if there's oxygen present, well, that's okay. We make the ATP. If there's not enough, now we're kind of stuck. Does that make sense? Okay, so I think I said in the first lecture, really a lot of bioenergetics comes down to, even not just bioenergetics, exercise physiology comes down to, is there oxygen present or not? If there is oxygen present, cool, we'll use aerobic pathways as long as we can, because we can make more ATP with aerobic mechanisms than anaerobic. But if we run out of oxygen, or there's not enough for the exercise intensity we're performing, then we have to shift to these other pathways that give us ATP a little faster. It's not as much, but it's available to run for our lives. We're running from a bear, right? It's like our body doesn't know we're running on a treadmill hard, but it just says we need ATP, so we're going to get it as anywhere we can. Cool? So the electron transport chain produces ATP via oxidative phosphorylation. That's where that term comes from. ADP is phosphorylated into ATP with the presence of oxygen because of those hydrogens are pulled through ATP synthase. Now, in some textbooks, you might actually see why we see the difference in, okay, look at glycolysis anaerobically. How many ATP are produced from glucose? Net 2. Glycogen, net 3. You'll see that in every textbook, right? Well, if you look at different textbooks, if you look at the aerobic contribution, you get 32 or 33, or you get 33 or 34, right? There's some fluctuation depending on which textbook you look at, because some textbooks don't take into account that when this NADH drops this hydrogen off into the intermembrane space, remember, it's actually it's pushing a substrate against a gradient, right? There's not a lot of hydrogens floating around here. They're actually all attached to NADH. There's a high concentration up here. So remember, if you want to push something against a gradient, that costs work. It costs energy, right? You got to do work to push it in there. So what happens is that you don't produce an exact 3 ATP per NADH because it does require a little bit of work to pump it into that intermembrane space, okay? So that's why you get 2.5 ATP per NADH. So if one textbook is using 3 ATP per NADH and one textbook is using 2.5, we can see that now there's a discrepancy because look at all the NADHs that are produced in metabolism, right? They start to add up over time. So that's where that discrepancy comes from. But ultimately, those hydrogens from these chaperone molecules are converted into water when they join with oxygen. Okay, that electrochemical gradient is what really drives that ATP production because number one, those hydrogens naturally do fall through ATP synthase, right? So even if there's not enough oxygen, they're still going to come through that ATP synthase molecule and produce ATP. The problem is that if you need a lot of that ATP very quickly and there's no oxygen, it doesn't happen very quickly, right? But if there is a lot of oxygen, those hydrogens come through a lot faster and you produce more ATP a lot faster, okay? So really, this driving factor here, right? We drop off these hydrogens, right? We move those electrons along the cytochromes and along the pumps. In this intermembrane space is a bunch of hydrogens. They want to pass through and form water. They also pass through ATP synthase. And if there's oxygen here on this side of the mitochondria, they want to be pulled through into this side of the matrix, okay? And in the process, you produce ATP. Now this is actually what ATP synthase looks like. Notice how it's kind of like a carousel, right? It's kind of imagine you were like on the horses, right? You're kind of like spinning around, right? So it's got this kind of rotor system, and then you have this kind of piece right here of it where I don't remember if it's the phosphate on this side or the ADP on this side or vice versa, but imagine there's a inorganic phosphate here and ADP here, okay? And so ADP, ADP, ADP in each of those. And imagine as this hydrogen comes through and passes through, it literally turns it so that, these form, they get pushed together, ATP. That hydrogen went through, but now this has turned one slot over, right? Another hydrogen comes through. So then this ADP forms with another inorganic phosphate, produces another ATP. And it kind of just keeps rotating along the way to produce the the ATP because the hydrogen come through. If they do not come through this protein channel, there's really not much to really force to really turn this motor, right? So if it's not turning, it's not phosphorylated. Make sense? And pretty much just stated all that, right? The purpose of the citric acid cycle is to produce all those NADHs and FADHs so they can go to the electron transport chain to produce the ATP. But remember, all those NADHs and FADHs, they are storing potential energy, right? They have not actually made ATP yet. They have the potential to make ATP in the ETC. So the idea is that we produce all these hydrogens and then they make the ATP when there's oxygen. Now, you don't have to remember the numbers of ATP and the NADHs. I'm not going to require that for this class, but you do need to know this slide. This slide has the rate-limiting enzymes for glycolysis for each metabolic pathway, okay? So, not just glycolysis, for every metabolic pathway, sorry about that. So we have each metabolic pathway, the rate-limiting enzyme for that, and then also we have stimulators and inhibitors. Okay, you have to know this for your exam. Like I told you, you gotta know the PDH slide, the phosphatase and the kinase will regulate that. I'm telling you that's on your exam, at least 3 questions. This is another one. It's a must know, okay? And we can look at this and say, oh, that's pretty easy to make. That's easy to make questions out of. All right, what's the rate-limiting enzyme of this pathway? Here's the options, pick one, right? It's very easy to do that. Which of these is a stimulator of this enzyme or of this pathway? Which of these are not an inhibitor of this pathway? Okay, so you've got to know like the terms and what stimulates and inhibits. But ultimately, if we look at the stimulators and inhibitors, we actually see a pretty common pattern, right? What is a common denominator? Look, ADP, AMP, inorganic phosphate, these are all signs that we've broken down ATP. So notice that if we've broken down ATP, these byproducts of ATP breakdown stimulate the rate-limiting enzymes to make more ATP in every single pathway. If we have a lot of ATP in the first place, that actually inhibits that rate-limiting enzyme. Why? Because the whole purpose of this pathway anyway is to make more ATP, and if we have enough of it, there's no need to turn it on. Okay? And also think about it like in some enzymes like phosphofructokinase, if you have a lot of ATP, it actually sits into the active site and blocks the active site of where AMP would go in and stimulate it, right? So if ATP is sitting on that active site of PFK and preventing it from being active, that's going to keep that enzyme off. You break down the ATP, that allows AMP to increase in concentration. AMP sits into the active site and increases the activity. And now PFK is highly stimulated, you make more ATP. Okay? But we see a pretty common pattern here with the stimulators and inhibitors. So, and practically, like an increase in calcium. Okay? When does calcium increase into the cytoplasm?
Muscle contraction.
During contraction. When muscles are contracting, they're using ATP. How do we make more of it? We turn on the Krebs cycle. We turn on isocitrate dehydrogenase to make even more ATP. Well, how does it know to turn on? Because there was calcium present that stimulates that enzyme to be more active. But we also know that calcium stimulates glycogen phosphorylase and glycogen breakdown. Right? So both those things are saying, hey, calcium's being released, there's muscle contraction going on. Okay, let's try to make more ATP from these substrates like glycogen. How do we do it? Stimulate the enzyme. Make sense? So, for sure, make sure you know this 100%. I'm telling you, there's 3 to 4 questions just from this. So if we think about that concept, the PDH enzyme activity, we said there's 3 questions from there. There's at least 3 questions from this. That's 6 questions, okay? That's already gonna be like, 10% of your grade just from this one singular lecture of material I'm telling you need to know, okay? So make sure you know those. All right, we are gonna switch gears and talk about cell signaling now. Well, do we have any questions about metabolism before we move on? I want to make sure you have any, if you have anything from that stuff, you want to answer any questions about it, you can go over one or two questions right now if you have any. Feel okay? Okay. One thing I'll say about the quiz, okay, if you get, if you get all the questions right on the quiz, good. I think you're in pretty decent standing, right? Pretty good standing. If you get one question wrong, I'd also say you're doing okay, right? It's totally, maybe that one question tripped you up. It's a 75 out of 100, but realistically, that's actually not that bad. If you get 2 questions wrong, I would probably be like, oh, this is like a red flag for me, okay? So if you get 2 questions wrong, yeah, a 5 out of 10 on a quiz, this is a sign that you need to refine your studying in some way. Either how you're engaging with the material, how you're studying it, how often you're studying the material, something needs to change. The exam is still like 3 weeks away, 3.5 weeks away, so it's enough time to get ahead of it, okay? So use this opportunity to say, hey, if you get a half on the exam, on the quiz, okay, don't take it to heart, like, oh my God, this is my life, it's like, okay, it's just a quiz, okay? You can come back from a 50 on a quiz, it's not a big deal. But it's just a signal, it's telling you we need to reassess what we're doing with the class, okay? So if you don't do as well, don't go into major stress, just say, hey, I need to reassess what I'm doing. If you do well, that's in pretty good standing, but don't say, oh, I know the material now, because the other stuff we're gonna go over is, up to this point, is still really good, right? So there's still 3 more lectures we're gonna cover for the exam. So, cell signaling. We are gonna talk about how molecules talk to each other and relates to adaptation. So we've actually been kind of talking about cell signaling already with bioenergetics, okay? There's a couple cell signaling mechanisms in terms of how your cells talk to each other and communicate information, so both within the cell and also with other cells or in distant tissues. So an intracrine signaling would be when you have one enzyme or protein signaling or talking or communicating messaging to another inside the same cell. Okay, this is not all that different than how we talk about enzymes of glycolysis talk to each other, right? Or PDH activity. This one phosphorylates that one, right? Which then causes a change in how substrates are metabolized. So this is not really that different than that concept already. Except this time we're talking about an enzyme activates another enzyme or protein and then turns on something in the nucleus to cause another nuclear or cellular response. But this all happens within the same cell. You also have juxtacrine signaling where this cell is sending a molecular signal to an adjacent cell. I like to remember juxtacrine as adjacent because they both have the letter J in their terminology.